you may be familiar with this figure from a paper that was published by Ann Hendricks's group back in 2017 in Nature Methods, where they looked at the EV literature that had been put together before this paper came out. So over 1,200 publications, and what did they find? They found that of those 1,200 publications, there were over 1,000 different separation protocols for EVs. So some of these were vastly different from each other. Some of them were fairly similar. But this indicated that there is maybe a difficulty in getting things to reproduce because we're using so many different approaches. And then in addition to that, they also found that there was inconsistent reporting in the literature. So sometimes the methods were not characterized well or reported well. And in almost 1/5 of the papers, there was absolutely no characterization of the EVs. So people were just saying we're working with EVs. They didn't tell us, how we know that they're working with EVs. In my lab, recently we've been doing some work with EVs in tissue. So this is a particularly complex source material, I think, for EVs, because you have so many different kinds of cells in the tissue and you have extracellular matrix. It's really hard to sometimes get at the stuff that you want. And so it's very important here to do a very gentle dissociation of the tissue so that you don't damage cells and create artifactual particles that look like EVs. And for the same reason, it's so important to do careful characterization. And when we looked back at the literature of people who had gone before us in doing some of this work, it turned out that many of the papers were actually grinding up the tissue. So putting it in a mortar and pestle or something and grinding that up, and then not looking at markers to understand if the if there were cellular artifacts there. And this is I'm not criticizing anybody here. I mean, I think there were some very pioneering papers here. But as time goes by, we want to see a progression towards more and more rigorous research that takes some of these considerations into account. Another great example of where we have diversity is this table that Rink has put together. And these are some of the variables that we could record in our collection and processing of blood plasma. So you look at this from the blood collection to the tubes that we put the blood into, the time and the different aspects of the processing of the blood, and then finally its storage. There are so many variables here. There are 40 that are listed here. There might even be more than that. So we have a lot of things to overcome. And then finally, before I go on, I just want to again talk to you about the physical properties of EVs. So we have our charge, we have our size, we have our density, we have our different surface markers, and unfortunately, there's a lot of overlap in some of these physical characteristics between EVs and other extracellular particles or EPs that might co-isolate or co-separate with the EVs. So we have a lot of challenges in the field, but I think we might be up to the challenge. Now, a question that Rink often asks is, what is the typical concentration of EVs in human plasma? This should be a slam dunk for us, right? We should be able to figure this out quite easily. But if we go back to some of the early literature on this concentration, we see that there is a difference of many orders of magnitude. I've even seen some published estimates that are higher than this highest one here. So even though now we're in the year 2020, we still don't have, I think, a good understanding of what that concentration is. If you go to a clinical chemistry lab, give them a vial of blood, they're going to tell you how much HDL, how much high density lipoprotein is in there, and they will be able to tell differences down to single digit percentage points, which is pretty amazing, and it's something that I think we would love to be able to do in the EV field, and someday maybe we will. I think we're making progress. So there was this wide range, and so we could ask ourselves if we have a crisis of rigor and reproducibility in EV research. Any field in science can ask itself this. So we have thousands of papers that are coming out every year. I think it's over 6,000 papers, perhaps last year, that looked at some form of EV or another. And we hear these great claims about EVs, and we want to buy into these claims. We want this all to be real, all the hype to be real. They're biomarkers of everything. They can be used to treat anything. But many of these studies are also single lab studies or single center studies. They have incompletely described methods so that I can't go in and repeat these studies. And as a result, the findings themselves are hard to compare. So with this in mind, I think we need to think about how we build a solid construction. So we want to start with the blueprints. Don't just start stacking bricks on top of each other. Have a blueprint. Get your scaffold in place. Do that homework, that legwork that you need before you start the project, or otherwise you're going to end up with this house that has collapsed. So thank you, Rink, for this wonderful illustration of what happens if you don't give the forethought to the project. And so I'm going to tell you about what ISEV. I mean, I think ISEV in some ways was founded um, in part to encourage rigor and reproducibility in the field. And so we want to make sure that our different studies can be compared. We want to make sure that we avoid overinterpretation. So do we know in this, my mouse study of a particular cancer type, is a given property actually due to the EVs, or is it due to some of the co-isolated components like the other extracellular particles? And we've even had several journal clubs over the past year that look at this issue. And then we also want to ask, is it really a certain subtype of EVs that are causing the effects? Often, one of the categories that's often brought up is exosomes. Are there really functional differences between exosomes, that is the EVs that come from the endosomal compartment, versus other types of extracellular vesicles? Or is it just that you have a bag of lipids and proteins and other stuff there that can be put onto cells? So do I need MSC EVs, or can I just work with milk EVs or urine EVs? So a bunch of questions that I think that we've tried to bring up in ISEV. And so the International Society for EVs, many of you on this call are quite familiar with it. But we have, various tools that we try to use to encourage the field and to encourage rigor in research. These include our flagship journal, the Journal of Extracellular Vesicles. We have various meetings. We have 2 massive online open courses right now, and a third is in preparation by Dr. Lei Zheng. We also have recently established this rigor and standardization subcommittee that Rink and Juan are co-chairing. So our first foray into establishing some sort of like community consensus guidelines or recommendations for EV studies was done by the ISEV board back in 2014. And so these individuals got together in one place and they talked about this for a while and came up with a draft. And so this paper was put out then at the very end of 2014. And this was the first MISEV, or minimal information for studies of EVs. And the gist of it was that we wanted people to characterize as much as possible the EV population that they were using, and then to demonstrate that whatever output they saw, whatever outcome they saw, was indeed specific to the EVs. And you can do this by doing a very specific separation process. This then led into another effort. We put out a survey, I think it was 2016. We asked people, what do you want to see in developing more guidelines or more extensive guidelines? And the overwhelming response was, we want to be involved. This is not just up to a few board members sitting somewhere in a room. This should be community-wide. And so we took the survey to all ISEV members, and we came up with this MISEV 2018 paper, which, I think a lot of people on the call will know this paper, but, I just gave a talk a few days ago, and it turned out that a lot of the EV researchers in the audience were not even aware of it. So we need to make sure that we're doing a good job of spreading the word about the efforts that we've put in together. And so the contents of MISEV 2018, we have nomenclature, we have collection and pre-processing. We basically go through all of the steps of an EV experiment, from the initial planning all the way to, what are your functional studies? What are your readouts? What are the controls that you need? And then reporting, how are we going to report our results? What's the best practice for reporting results? All right, so another thing that ISEV has done is to monitor the patient. So if the patient is the EV field, we've tried to take a pulse of this field every once in a while to see what's going on. And an initial attempt at this, which I think has also been a very, very well received paper, was led by Chris Gardner, and this was called Techniques Used for the Isolation and Characterization of EVs. So we started a new survey. We tried to see what has changed in the intervening years between 2015 and 2019. And as a result, this methods for separation and characterization paper was published last year. There was an accompanying editorial also that came out in JEV just a few weeks ago. So, who participated in this survey, and what does this tell us about involvement in the EV community? Well, we actually had about 620 responses at some level, but 320 of them were complete. So people had gone through all of the questions and answered all of the questions. And this was approximately 2 times the engagement that we had in the Gardner et al. Survey from 2015. We also noticed that there were 40 countries that were represented on 5 continents. So this was truly a worldwide response that we had. And we observed a large increase in representation of several countries, including China, Korea, Australia, and then in Europe, Italy and Portugal had really come up in the numbers. So 85% of the people who responded here were ISEV members, and 86% were in academia. About half of the people who responded were principal investigators. So this shows us that there was, people who were leading labs were very interested in contributing their experience with EV research. The next question that was asked in the survey is what is it that we're working on? And overwhelmingly, most people are working on cell culture conditioned medium. So people are doing cell culture experiments, and they may be using medium without serum, or they may be using medium with serum. So there are more people who are using serum-free medium than serum containing medium. And in fact, that proportion went up significantly between 2015. So you see the kind of light yellow bar there, light green bar is 2015, and then above it is 2019. There was a significant increase in people who were using serum-free medium. The other sources that you see here are also quite important. So there was even an increase in people who were working with blood plasma and blood serum, and others like urine increased, tissue. I mentioned tissue early on. That was not even registered in the previous survey, but now there's a good number of people who are working with tissue as an EV source. So next on to that third category in MISA, which is EV separation or isolation or concentration. So not all methods are actually separation methods; they might just be concentration. In this snapshot article, which is a really nice, very brief and in-depth overview of the separation techniques right now, that was published by Clotilde Thierry's group last year in Cell, you can see something that MISEV also recommends. So of all these different methods, each of them will fall somewhere on what we call the recovery versus specificity matrix. So recovery, of course, is how much of your input, EVs are you getting out at the end, and specificity is how specific is that population? Did you get just your CD63 positive EVs, or did you get just your large oncosome population? And so each of these techniques is going to fall somewhere on that matrix. And what we recommend, what MISEV recommends, is that you need to tell us, why did you do this technique? Okay, you did PEG precipitation. Why did you do that? Or why did you do asymmetric flow, field flow fractionation? And where, what were you trying to achieve? Was it maximizing yield, or was it getting that high specificity? So what were people actually doing in the survey that we put out? Well, it turned out that I think the Gardner et al. Paper is often cited by people who use ultracentrifugation, and they say, well, we still use ultracentrifugation because it is the most commonly used method in the field. And that continues to be the case today. So ultracentrifugation, stepped ultracentrifugation, or differential ultracentrifugation is still the most widely used method in EV laboratories. The use has apparently declined a little bit percentage-wise, but that was not a significant decline. So I don't think that we can say for sure that it has gone away at all. So I'm just going to stop here for just a second. If you'd like to ask any questions, please put those in the comment box. As soon as we're done with this presentation, then we'll unmute everybody, and anybody can jump in here and discuss. And Juan and Rink too, if you have any comments on any of this, please interject. So these methods, we can look at these methods and see a few interesting trends. So the size exclusion chromatography, for example, had a very big increase and significant increase, as did gradient ultracentrifugation. So trying to separate things by density using the ultracentrifuge, that increased. But we also saw that there were several other techniques that increased, like precipitation. And I think I was most surprised by the result with combinations of techniques. And it's almost as if the decline in combined techniques is probably explained by size exclusion increasing, and that's been my interpretation anyway. That a lot of people have moved to just using size exclusion chromatography. And some of these techniques, it's interesting also, you could put them into the category of gentle separation. So size exclusion, you're just doing usually gravity flow through a column. You might have you might have your filtration technique. So if you don't put a lot of force on that, it can be a very gentle technique. Precipitation, obviously, or affinity, these also might be very gentle techniques compared with the very high G forces that you're putting onto a sample using ultracentrifugation. So we seem to see a move towards more techniques that are relatively gentle. And think a slide that Rink also put together was that, when you look at past studies, most of them were using differential centrifugation to obtain their population of EVs before they did their assays. And in recent studies now, I think consistent with this survey result, we see that size-based separation, just size-based separation, which could be size exclusion chromatography, it could be tangential flow filtration, could be AF4. These are really coming into an increasing use. And and yet, I think we want to remember that combinations of techniques are often important if we really do want to be sure that we're getting a pure population. So these might be combinations of density and size. It might be using several different sizing techniques so that you get the right range. It might be that you do size and charge. And so on. So each of these techniques that we've mentioned has a place, I think, in our armamentarium of EV separation technologies. We just have to understand why we're doing those techniques and what the effects are. So some interesting, I think, observations about trends in the field. Now, so we've done our techniques. How do we know if our separation actually worked? Well, here's Schrodinger's little kitty. It's alive and dead at the same time. And we don't know if those EVs are there, if they're contaminated or not, until we open the box. We got to open the box and do that characterization. We've got to ask the questions like, how many are there? How big are they? What's on them? What's in them? Are they squishy? Where did they come from? And do they work? Maybe we want to do some functional assays too. So in our survey, what were the items of interest, so the molecules of interest to our survey respondents? Proteins and RNA were clearly the ones that most people were most interested in. But I also know that there is an increasing interest in some of these other categories, including the lipids, the metabolites, and the number of biomolecules that different labs are looking at. Most labs are not just looking at one biomolecule. They're not just RNA labs anymore. They're not just protein labs. They're looking at multiple of these biomolecules. And indeed, it can be difficult to say a lot about a population if you only look at one of these molecules of interest. So to look at those molecules or to understand about the EVs as a whole, what are people using? The Western blot is still the most common technique that people use to try to characterize their EVs. So of course, a very powerful technique. It can also be a very hungry technique. It's going to take a lot of material. But Western blot continues to be very used by almost all labs, really. Single particle tracking is just behind there. So being able to look at a population of particles and see how they, for example, scatter light, so that we know a little bit about their size and maybe about how many there are. We can count those single events. protein concentration is also used quite a bit, and even more than in 2015. And then you see that there are a bunch of other techniques. You could probably come up with techniques that are not on here. So we have others down at the bottom. We don't have everything on here. But I just wanted to also mention this box with the black bars on the side. In 2015, most of our respondents were saying that they used 3 methods or fewer to characterize their EVs. But now the majority of our respondents are actually using 4 to 6 methods, and some are using even more than that. So we see that the field has moved towards more of multi-assay kind of approach to EV characterization. What about if we separate things by bulk analysis versus single particle analysis? So we could go down this, go down through each of these techniques, bulk as B, SP as single particle. So some of these techniques can be bulk or single particle, like flow cytometry. So if you're capturing EVs on a big bead, and then you're just looking to see if there's positivity for something, that would be a bulk EV analysis. But if you're using flow cytometry to look at single particles and phenotype them, or not phenotype them, just count them, that would be a single particle analysis. And so we see that there are is a good distribution actually of bulk analyses and single particle analyses. Something new that was done in this survey that we hadn't had in the 2015 survey was to ask some questions about biobanking. So if you've recruited patients, for example, for a study and you're collecting their urine, you might want to store that somewhere under a very strict SOP so that you can go back to it later and be sure that you can make comparisons between groups, between different cohorts. Biobanking, we asked the question of whether people are doing biobanking or not, or if they're getting samples from biobanks, and whether this had some association with some other ways that you can approach your data. So, did you do biobanking, yes or no? And then, did you do some form of quality control? Did you assess recovery, like after you got your EVs back? Did you do something to understand how many you were getting out, how pure they were? And then finally, do you do normalization? And I think across all of these different questions, you can see that the people who were doing biobanking or getting samples from biobanks were paying a little bit more attention proportionately to these questions about quality control. And maybe not so much with normalization, but definitely with the prior quality control and with the recovery estimates. So it's almost like there might be a mindset, if you're thinking about something according to an SOP for biobanking, you're probably also likely to be considering, how do I measure how well this biobanking went? How do I assess, how well my experiments are going? So I thought that was kind of interesting as well. So a final question then is, okay, we normalize. How do we normalize? How do I know how many EVs or what quantity of protein or whatever I'm going to put into my in vitro assay or into my mouse model? So here we divided people into groups based upon whether they said that they did in vitro assays, in vivo functional assays, both, or actually they might be doing normalization for other things that were not functional assays. So we found here that for the in vitro studies, when people are taking EVs from one source and then putting them into their cell culture system, it was EV number or particle number, perhaps, that was the most often used, followed pretty closely by protein concentration, cell of origin number. So you can say I got EVs from 10 million cells, and then even like starting volume, which is sometimes you have to define that very carefully. In contrast, for the in vivo assays, it looked like there were a lot of these different categories were about the same, but there was a big difference between the in vivo and the in vitro. And then for both, it looked pretty much like it did for the in vitro, the in vitro assays. So that was, I think those were the findings that we had for the normalization. So that was pretty much the results of that 2019 survey. And I just want to mention a few other tools and initiatives that that have been supported or endorsed by ISEV. And one of these is EV Track. So I showed you some illustrations from the paper from on Hendrix's group in 2017. And they set up this EV track system where you can go into that system, you can go into that knowledge base and deposit the technical details of your experiments. You will then get a kind of a score back. And this is something that can be useful for coaching you. It can indicate things that you can change about your experiments or that you can add to your experiments. In some cases, even just details that you need to add to your methods section, like which antibody did you actually use? Which rotor did you actually use for your differential ultracentrifugation? So that you can improve the quality of your reporting and potentially even the quality of your study by trying to get as much in there as you can. So that's a very useful tool. We've had other things that have come out, the flow cytometry consortium that ISEV members, along with the International Society for Advancement of Cytometry and the International Society for Thrombosis and Hemostasis, have been working on since back in 2015, when they first came together at the ISEV 2015 meeting. But this particular guidelines article was initiated in 2017, and then it was published early last year. So this has a reporting framework that you can also follow to make sure that your studies with flow cytometry and EVs are going to be reproducible and we hope believable as well. And then I'll just conclude with this note on MySev and ongoing efforts that are related to MySev. So we have this rigor and standardization subcommittee. There are, I believe, what is it now, Juan and Rank, 10 or 11 task forces, something like that?
I think it was 10. That's
10 task forces now. So these, we have a process in place of how people can apply to start a task force. So then there's a review process. We want to make sure that that there's a critical mass and that we have the ability to recruit the experts to work on these task forces. But one of them, so Aled is on here today. He led the blood EV roadmap paper that was written after a workshop in Birmingham, UK. We have some reference materials, a reference materials manuscript that was published by Josh Welsh. So Josh and Ann Hendricks are leading our reference materials task force. We have a regulatory affairs and EV therapeutics task force that has put out recently some recommendations around COVID-19 therapies and EVs. And then we have several task forces that are devoted to specific sources of EVs in addition to blood, like urine, milk, saliva, CSF. We have a bacterial EV task force. So please stay tuned for developments from these task forces, which I think are going to nicely complement what MySev has achieved. And feel free to reach out to any of us if you would like to talk about maybe a topic that you don't see represented and that you think should be. And of course, we're going to be updating MySev. So MySev cannot be a static document. It's not something that was written 300 years ago and will stay the same forever. This is something that will be updated over the next few years. So this is, I think, an overview of some of the things that ISEV has done with rigor and standardization recently. And I'd like to, this is not my work. I've contributed to it, but there are many people in the society who have helped to make, I think, the ISEV endeavor something that is useful to the community. And I think it has had influence. I'm sure that it has had influence. So thank you all for your participation as well. All right, so any questions, comments, complaints? I'm going to stop sharing, and I think Rink and Juan will hopefully help me with answering some of these questions, and anybody else in the call who'd like to jump in. I'm going to allow unmuting now. So thank you very much. So what do we have in the chat box here? Desmond Pink, do you want to start?
Sure. Just want to make the comment that a lot of these enrichment processes are time consuming. And if you're building your EV research, your exosome research from the perspective of finding biomarkers and validating them for a specific disease, and you want to take this idea to clinic, you also have to consider the scale-up process, including validation, reproducibility of any of these processes. That takes a lot of time and a lot of money. and a lot of forward planning. So I think that needs to be built into this. I mean, all of those things fall right into the path of rigor and reproducibility.
Yeah, and this is one of the task force, for example, that is taking into account how to produce how to purify the vesicle from condition median, taking into account for this, for example, use therapeutic or this kind of thing. The idea with this task force is to try to compile what will be necessary to fulfill the requirements that will need, for example, this. In the case of the therapy, for example, is the one that is more focused at the moment, okay, the initiation of this task force.
Yeah, no, we've had the similar experience for diagnostics. It's a question that I get asked almost daily: how do we purify? And we've actually at Agnostics taken the approach that we don't purify for the simple reason that it adds too much variability into the process. Also, it adds an additional amount of time and money on the diagnostic floor that the technologists and the different diagnostic companies aren't willing to undertake right now.
I think that's a great point, Desmond. Especially in diagnostics, the ideal situation would be that you don't have to purify at all, that you're able to identify these particles without any separation process. Yeah. I think that's the goal, that's a goal that we should have, if when it is possible. Of course, if what we're doing is taking a bulk population and measuring a particular molecule, we would at least want to in advance show that if we do purify it, that molecule is associated with the EV specifically. And once you've done that, then maybe it's not important to purify. So it all just depends on exactly what you're trying to do with that diagnostic test. And similarly in therapeutics, there are many situations where the release rate itself is what is giving the value. The EVs may contribute to that, or they may not. And it's, again, for the basic science side of things, you might want to know what is that component that's contributing. But ultimately, what we want is to help the patient. Yeah. And if we can do that with a less purified population, then so be it.
Well, I think even prior to the science part of it, you have to ask yourself the question: if I'm going to process 1,000 samples every week, how many ultracentrifuges do I need to have to actually do this? And can you actually manage the throughput? I mean, I think a great example is, the rollout of the vaccines. The diagnostic centers or the different places, they didn't all have -80s. I mean, I think that's a really crude example, but it hits the point that not all of these sites will have some of the technologies, as a freezer we're talking about, that we sometimes as researchers take for granted.
Desmond, maybe I can add that I think I agree with you. In Amsterdam, we like to measure the vesicles directly in the body fluid. And as Kenneth showed from all slides from me, we have long been struggling. What is the concentration of vesicles, let's say, in normal plasma? It looks a simple question. The answer is difficult, but I think within 1, 2, 3 years, we can reproducibly measure the concentration of vesicles in plasma. So an infrastructure is being built with traceable reference materials dedicated for flow cytometry, for example. We're in the process of developing stable biological test samples. And I think next year we will do an interlaboratory comparison study where we calibrate instruments, flow cytometers in different labs, provide the stable biological test sample, which has also been characterized by traceable measurement techniques. And then I think we will be quite far in being able to measure directly in samples concentration of, let's say, cell-specific vesicles. So it's a matter of time, I think.
It's a point that we've struggled with from day one, and we've talked to the different regulatory agencies. So what we've done is we've built in our own controls, and we've made sure that our SOPs have been very standardized and followed and translated into a diagnostic site where, as a researcher, we developed the protocols and then we sent it to a second site, and we've done the comparisons to make sure that, I think, Ken, you had one of the slides, like we've built something and then we've checked it to make sure we're getting what we think.
Yeah,
and that's crucial.
Yeah, but I would like to go one step further, and that is, if I measure a sample or you measure the sample, that we find the same thing.
Yes.
And that's something that I think within 2-3 years we can achieve, at least with flow cytometry. So I think that's clinically very important, because I think it's a prerequisite that we measure the same thing, and then we can study whether it's relevant and differs in the disease or not.
Yeah, I will tell you though, and I think it comes back to the point that Ken made at the beginning. These types of experiments, from the perspective of a lot of PIs that I've dealt with, they're not sexy.
No.
Flat lines, don't make grants, but
it's boring.
It's boring, but I tell you what, when I can show you a year's worth of data or, controls that we've built, and I can show you that they're flat over a year, I trust my data. But more importantly, the big companies that want to take these ideas and actually use it as a diagnostic or a therapeutic, that's the data that they want to see.
Yeah.
That is really the data they want to see is flat lines and recoveries.
Great. Well, thanks for those insights and comments, Desmond. Much appreciated. We have a functional assay question here from, is it Chezi?
Yeah, I have a question related with this functional assays. My question is if we are, is necessary to assess the function of non-EV components, for example, those that are co-isolated with the vesicle, to prove that the given biological function, for example, is related with the EVs, or is related with these components altogether. My question is, we have to do this with all the functional assays?
Well, I guess it just depends on what you're trying to claim. So if your claim is that EVs are causing this particular effect, and you've done, let's say, PEG precipitation, and you put this into your system, that would not allow you to claim that it was EVs that exerted the effect. So you would want to instead start with some sort of a system where you're taking different fractions. So for example, if you put it through size exclusion, you use your EV enriched fractions, you use your EV depleted and protein enriched fractions. Or you might go, as you said, sucrose gradients or iodixinol gradients. You might want to look at different. So the key is to understand if the EVs are really essential for this activity or not. If you want to say that it's an EV mediated therapy. If all you want is to see an effect and you say that's the cell release rate, then it's not necessary, right? It's just about what you want to claim as the mode of action.
Okay.
Any other comments on that? No.
I just would add that sometimes not function could depend also. I mean, could be a balance between the vesicles and the contaminants. So you need to have, maybe not. Sometimes it's not good to call contaminant. Just associate to the go to the vesicle, because you have to think that in the biology is not like you have everything isolated. You have together and work together, and the physiology is complicated. So sometimes you have to consider both. The point is that it's difficult at this moment to play like I put 10,000 molecular exosomes, and then 10,000 of the other molecules that could be contaminant, and this has the functionality that you are looking for. So at this moment, we need to be open to the different possibilities. That and if you see an effect with the vesicle as more as pure as possible, okay, this will be your follow to point, and then you can go with trying to contaminate with the associates and see how this influence the function. So this is my point, is be open to a possibility that the vesicle alone is not the one that is doing the function. So.
No, and I think maybe I can add that there were recent papers that corona proteins can be present on vesicles. So the surface of vesicles is surprisingly large, and proteins from the environment can bind to the vesicle surface. And the other thing is that if you work with vesicles from blood, lipoprotein contamination often is a problem. But platelets, for example, and platelet vesicles have receptors for LDL particles. So part of the vesicles of platelets will be complex with lipoprotein particles. So you simply cannot get completely rid of the LDL particles when you isolate the vesicles from blood. I mean, it's just a complex input. So yeah, as Juan said, it's really complex. It's not as easy that you can separate the lipoprotein from the vesicles if they're complex.
Yeah, that is a problem we have in our laboratory. Thanks for that.
No, you have a challenge. You don't have a problem.
Yeah, we have this problem.
I think, Rink, this comes back to your earlier question about quantifying the numbers of EVs in plasma or serum, whichever blood film you're looking at.
Yeah.
If you monitor, and we've done this in the lab as a sort of an exercise, take a fasted sample in the morning, and then go down, just have a simple cup of coffee with cream, then have a donut, and just watch your concentration explode.
Yes, but what is exploding is, and we actually, that's interesting. For example, Frank and Edwin have had a fat breakfast in the morning with a lot of cream and eggs and that sort of stuff, and then we have blood fasting and fat. But for example, with flow cytometry, we are now able, based on refractive index, to separate fat particles without labeling from vesicles. Yes. And that's a very powerful trick. And indeed, what you see is that there's a massive increase in fat particles. But as long as we are sure that we can discriminate them from the vesicles, I don't bother at all. So I'm also not interested in whether it's a fasting sample or not, because in the clinics, it's usually impossible to get fasting samples.
And I think that also comes back to when we make criteria as you're making your assay, you're developing it, you have to think, what is the standard procedure that is going to be followed in a test? So for us, I mean, we get plasma samples or serum samples rather for prostate cancer, and those aren't fasted. So we have to deal with non-fasted samples, and that's just a reality. We're not going to change the standard of care for that procedure.
But for example, one of my interests already for a long time is measuring the association between the clotting activity and vesicles, and tissue factor is only present on vesicles if you really remove all cells. For example, ISEV is scheduling a standardization study, interlaboratory comparison study next year. But for tissue factor activity, there are biological standards available from the industry. So I think there you can quantify the clotting activity associated with the vesicles to the international standards that are available. So that's quite, that's an easy, low-hanging fruit, I would say.
Good. We have a few more questions here too. So John Sperduto, you have a few here.
Yeah. So first question is a concern regarding how Ken mentioned that really it seems that the predominant mode of isolating is just one technique. But having a background in bioprocessing and transport mechanics, there's really not one size fits all technique, and there never really will be. In industrial bioprocessing, as a guide, you have, techniques that are suitable for clarifying, techniques that are suitable for isolate or for filtering and concentrating, and then you have techniques that are more suitable for polishing of the components, getting from that 90% purity to almost 100% purity. And I think there's a bit of a lack of consensus on where different techniques sit on that continuum. Obviously, some techniques can be good for both clarifying and filtering, like UC and TFF, whereas others, like chromatography, are much better suited for the polishing application. So interested to hear your thoughts on that. And also a similar concern is being a PhD student myself and trying to understand the nuances of each of these techniques, like UC versus TFF versus UF. I've had a hard time trying to find the underlying mechanics governing, what are the consequences to the EVs at the molecular level when you put them through UC, which is, pretty violent, versus say TFF, which is much less violent. And does that matter? Does it not matter? And then finally, one thing that I've also noticed seems to be a lack of use of SEC, or just general low pressure chromatography. to isolate, the number of, or to characterize the number of particles versus whatever marker you're looking at, be it total protein load, a specific marker, et cetera. And I think that would be an excellent standard to set for basically saying, all right, where our particle curve seems to overlap very strongly with our biomarker protein curve. So chances are, if we have EVs, they are associated with these proteins, since they would elute earlier in the fraction. That was a lot. So I appreciate any thoughts that you can provide on any of those questions.
So far, what I can tell you regarding that is that all the people, all the groups that has been comparing different methodologies, all arrive to the different by proteomics or lipidomics. There are changes. I mean, they aren't the methodology. Each methodology provide different population. Sometimes could be that is not the population the difference is something that maybe is what you say is the mechanic could influence and binding and broken and generating new artifacts or new type of vesicles. So far, I think it's not it's something to have in mind, and is people is already the different groups working on that. Nothing to say, but it's true that also previous years, the technology to characterize the vesicles was maybe not so sensitive, for example. And with the advance of the technology, for example, proteomics at this moment today you don't need to match to detect to do a good proteomics. So you can go and try to use less invasive and less more pure, like size exclusion chromatography, let's say, to apply that. So I think the combination came from the how was the technology to characterize what amount you need of material with what we have, the technique that we have for isolate. So, but yes, it's something to have a good variability of the different techniques, and these produce different yields and populations.
I think there's also not one answer. I mean, all fluids are different in composition, viscosity, whatever. And a slide that I think I also gave to Ken, but it didn't show it, but I like it very much. That's why I tell it now. If you take the size distributions of vesicles that have been reported, and you estimate quite a high number of vesicles, and I think it's quite realistic based on flow cytometry, you can calculate the total surface area of the vesicles per milliliter of body fluid, and you can calculate the volume. Now, the surface of vesicles in 1 ml of normal human plasma is about 50 square millimeter, which I found surprisingly large, but it is because you have so many small vesicles. But the total volume in 1 ml is 5 nanoliter. So the total vesicle volume estimated in 1 ml of normal human plasma is 5 nanoliter, meaning that you really have to try hard if you really, in the end, only want to look at what is in those 5 nanoliter. The chance that you end up with contaminants is huge, especially if you start working with complex fluids. So I think that there's not one answer, but I think that really shows how difficult it is.
Yeah, that is an excellent point. Something I've long considered, because it's a hard problem. Yeah, it's a very good point.
Yeah, and whatever we say about it, I mean, it's a young field. I think everybody's now trying to find methods, combine methods. And as Ken showed, I think that's nice. Vesicles have a few properties. I mean, they have a size, they have a certain protein composition, charge, whatever. And you can use all these different properties to isolate vesicles. But of course, it will depend on what your interest is and what your starting material is. But I'm pretty sure you will need, a combination of methods to get real pure vesicles. That will be really a challenge.
Good. Okay. So we have quite a few questions, and there are still some coming in. So if we're not able to get to everything, I will distribute the text file of the questions, and we'll see if we can contact people individually to answer questions. But the next one, and I'm just going to read this myself, Will, I hope you don't mind. Will Hotham asks, if we have characterized EVs from one experiment, let's say, or from a cell line, when we do another experiment with that cell line, do we have to go back and do the characterization again, or can we just refer to our previous protocol or our previous paper?
I think that some characterization of the preparation that you are testing in that experiment, you need to do it. So, because as we are talking about, the variation from batch to batch is still huge, and still we don't have the complete parameters to control that. So I think, yes, will be some characterization. This is my point.
Do you agree, Rink?
Yeah, very much.
Got to make sure that it's the same thing is happening all throughout.
Yeah, but if you culture cells, I mean, if you're 3 cycles further, are the cells still the same? And you have no clue at all. So I think every time you need to do your controls.
I think it's like running your protein standard curve.
Exactly. Yeah, it's
every single day.
Shitty. I mean,
yeah, and so there are going to be studies,
control,
though. But you're going to have studies where you, like I think especially with some of these biofluids where you don't have a lot of material, like cerebrospinal fluid as an example, you may not be able to characterize every single sample to the degree that we would like. And so in some of those cases, you've just got to show that you've got to show as much as you can. Like you might take a pooled sample, show that your technique is working for that pooled sample, and that might be the best you can do. But if you have the material available, I think it's important to make sure that you have some kind of a measure anyway to know that it actually worked.
We measure some like a cell lines, different cell lines. The marker is completely different. So you can see one cell line, the biomarker is there, but not another cell line. So particularly for different cell line, you definitely need to remeasure that thing.
Yeah, for sure. I think you can't just assume that a marker that you found in a paper for one cell line is going to work for another one. That's a good point. I think the last question then is going to come from Patrick James. So Patrick, did you want to? You had a couple here actually, but I think we can get to these, and then we'll try to address the rest of them offline.
Thanks, Ken. Well, I guess the first one was a response on my thoughts to Will. I think it lines up with what Ryank and Desmond have said about a minimal bridging experiments, if you want to. And I think Verena presented Berndt's work a couple of weeks ago, which sort of demonstrates quite nicely how different cell lines produce quite different effects. But I guess my question was more off topic, and I sort of came up with one of Ken's slides, and coming from where, so I work for a biopharma, is with the regulatory committee, have you got someone from the FDA or the EMA? Because some of these what we're discussing here, characterization or minimal requirements, some of us, and Desmond sort of raised it as well, some of us are targeting towards a therapeutic. We want to get an IND. So has there been any feedback about their expectations of what we should do?
Yeah, so indeed, that committee, that task force that was set up does include a few people who are in regulatory agencies. And then we also have established ties to others who are not on the task force, but who are very interested in the topic and who have given us feedback. And so we've also tried, we've also started a campaign to make that expertise available to regulatory agencies that might be looking for some advice on what is specific to extracellular vesicles as compared with some of the other things that they usually work with. So we're that's an excellent question, and I think we're working on it. But we'd always love to have more contacts. So if anybody has some contacts or knows of interest from regulatory agencies, please feel free to reach out to the standardization committee, and we'd be happy to put those contacts in the right places.
We are in this point. The task force is also working together or in collaboration with the ISCT, that is also with the cell therapy, and this has also different or similar challenges. And we are in this task force with this collaboration and this be together, let's say that. So this is a good point.
Yeah, and I think also the other point is different. So we've used a lot of consultants for our development process. Different consultants will have, of course, different opinions. And it's almost like, the heterogeneity of techniques here the heterogeneity of validation techniques will be similarly proposed.
Very good. Any last words here? I think we need to wrap up because we're at the end of the hour. Rink, Juan, anything else you want to leave us?
No, I thanks for the invitation. I thought it was a very nice presentation, very nice overview. And I think it's a very important topic. I think if we want to have some credibility of EV research, I think this is a good instrument.
Yeah, no, I agree. I think the idea of rigor and reproducibility is going to be hammered home continually. Every new generation of scientists that comes along.
I just wanted to say that this rigor and standardization committee is a platform for all the members of ISEV to be part active. So it's also I think is here. We are now setting is the starting point, but the idea is to stay here forever. Let's say in ISEV, and it's a platform for you and for all the members to be part. I mean.
Yeah. And I think it's also important, like industry is watching. I mean, scientific experiments are going to come, or scientific discoveries are going to come from wherever in the world, but we're always watching. And one of the first questions we ask is this reproducible? All
right. Well, thank you all for your contributions today, your thoughts. We will distribute these remaining questions and try to get back to people. But let's go back out there and make sure that we've got the blueprints right and the scaffolding right and get this built the right way. So, thanks everybody. Take care and hope to see you again soon.